My name is Huy Ding. I'm an assistant professor in Mercado, and it's my great honor to moderate the last scientific session of the symposium. Our first speaker gonna be Dr. Vinay Pathak from NCI. Before his current position and previous tenure at West Virginia University, Dr. Pathak got his PhD from UC Davis and did a postdoc here with Dr. Howard Temin from 1988 to 1991. They published the first reports in PNAS and Journal of Virology of the Z2A hypermutation in retrovirus, which is now known as many of you know involved in apoptotic enzymatic activity. The process, as we know now, contributes to several mutation signature in cancers. His current research focus is on studying different aspects of early HIV replication, including HIV-1 core nuclear import, encoding, nuclear trafficking, and integration site selection. And please welcome Dr. Pathak, and he's gonna share with us a talk titled "A Newly Revised Model in the Early State of HIV-1 Replication." Okay.
Thank you so much. Can you hear me okay? Great, okay. Okay, well, so much has been said about Howard's legacy by several speakers here already. John, Michael, Eric, Lou, and others. So I'm just gonna maybe reiterate a couple of points that I thought were important, and then I'm gonna share some personal memories of my time in Howard's lab, and then I'll tell you about some recent work we've been doing for over the last 5 years or so. And I'm gonna try to, pointer. There we go. Okay, so, and I'm going to try to stick to the script because I'm scared of running overtime, okay? What can you say about Howard's legacy? So in my view, perhaps his biggest contribution is that we knew about reverse transcription and retroviruses before HIV came on the scene. And we would not have known how HIV replicates when the AIDS pandemic began if it wasn't for Howard and David's discoveries. The first set of anti-HIV drugs were all RT inhibitors, and therefore, without knowing about reverse transcriptase, development of antiviral therapies would have been delayed perhaps by several years, perhaps costing millions of lives. And Howard worked on retroviral genetic variation for most of his career and was very interested in it. And we worked on the reverse transcriptase, the high error-prone nature of reverse transcriptase and mutation rates, which generate high genetic variation and leading to high evolutionary potential of retroviral populations. And this high genetic variation leads to antiviral drug resistance and makes it very difficult to develop anti-HIV vaccines. And these are two problems that we're dealing with even today after 50 years and are likely to be dealing with 50 years from now. Just my personal memories with Howard. So this is one of my favorite pictures of Howard. As has been said, he rode his bike to the lab. So I joined the lab in 1988, and at that time, Howard wanted me to work on spleen necrosis virus mutation rates, and wanted me to use a reversion assay that Joe Duarte had developed earlier. And by the way, it took me a long time to call him Howard. When I was in the lab, he was always Dr. Temin. It was really only after I left the lab for a couple of years that I sort of got used to calling him Howard. He wanted me to, although he wanted me to use this reversion assay, I wanted to do something different and develop a forward mutation assay using a lac repressor as a reporter. Yeah, okay. So he allowed me the freedom to develop this assay, which kind of took a while to construct the shuttle vectors. And he was very patient. And even after constructing the vectors, I realized it didn't really work, and I had to develop a method to actually purify profile DNA from infected cells to get it to work, and that took some time as well. Oops, sorry. Back. But anyway, so throughout this process, he was very patient. And once the assay started to work, he was just 100% behind me and supportive, and was often as excited as I was as we were getting results. And very often after the ritual good morning that Michael alluded to, that everyone got a good morning, he would often sort of hang around and linger and hoping I would volunteer some data or some information I had, because he didn't want to wait till Friday's confession to find out how things are going. So at the time of the publication, our paper was rejected by Cell, and much to both of our disappointment. And I wanted to publish in PNAS, but Howard said that I have too much data and it wouldn't fit in a PNAS format, and therefore we should publish in the Journal of Virology, which I didn't want to have happen. So it was sort of so I argued with him a lot, and I must say it was he was kind of mad at me for a while because I kept arguing with him, and it was mostly because of his frustration, because he also wanted the paper to be highlighted and didn't know what else to do. But so I argued that my only crime was that I had too much data, and therefore I'm going to end up being punished because of it, and get published in JP. And so the next day, Howard agreed to submit to PNAS, and we divided up the data into two papers, and they were published back to back in PNAS. And to my knowledge, that's the only time before that he's ever done that, to have two papers simultaneously in PNAS. So some other personal memories. So Howard used to have a July 4th barbecue at his home, and he used to invite the whole lab over. And once he invited us, and I informed him that I won't be able to come because my mother and sister were coming to visit for that weekend. And he very nicely, in his unique way, insisted and persuaded me that I should bring them along to the party, and he wouldn't say no, so I did take them over, and he made sure he talked to them both and made them feel very comfortable and welcome. This to me is kind of a funny story, but one weekend he had ridden the bike to the lab. I think I may have been the only person around, or maybe one other person. And he told me that the brake on his bike was broken, and he was very proud of the fact that he had fixed it. He said, well, it was broken, but I fixed it. And I did not, I repeat, I did not tell him that he had the Nobel Prize and no one in the world would doubt his ability to fix the brake. But this is just one of many examples of Howard taking the Nobel Prize and the fame in in stride and remaining firmly grounded. And it's the sort of the human side that I really love that I think Lou mentioned. And so Howard will always have my deep respect and admiration for his intellectual courage, nurturing of students and postdocs, and his commitment to social justice. All of these things have been mentioned earlier, but that's and many other enduring qualities. So I'm going to switch to telling you about some recent work we're working on, doing on the early stage of HIV replication. So this diagram shows what was the traditional view of HIV replication for until recently, for almost 30 years. I believe John could correct me, but when the virus fuses with the plasma membrane, at the surface, the core consisting of the capsid protein gets deposited into the cytoplasm. And it was believed that the capsid rapidly disassembles. Then the RNA is reverse transcribed into a double-stranded DNA. And that DNA, along with some viral proteins, get imported into the nucleus, where it integrates to form a provirus. There were a couple of labs who had also suggested that the capsid docks at the nuclear pore, unquotes, while it's docked there. But the main point was it was happening in the cytoplasm at this side of the nucleus. This was thought to be the case largely because the viral cores were thought to be too big to fit through the nuclear pore protein complexes. So the wide end is about 60 nanometers, and the nuclear pores were thought to be about 40 nanometers for many, many years, until just a couple of years ago, where that was revised to say it's now 64 nanometers, and making it possible for the capsid to enter the nucleus. So the other thing is, if you can observe capsid disassembly in the cytoplasm in biochemical assays. And this process has been difficult to study because quantitative assays to determine the amount of capsid protein associated with these complexes were not really available until recently. And the other complicating issue is that there are many viral cores that enter the nucleus, but only few of them go on to complete replication and integrate to form a provirus. We estimated that this was about 1 in 50 cores that enter the cytoplasm will lead to a provirus. So most of them are on a defective, non-productive pathway. And therefore, biochemical assays are going to give you the average of what's going on with the bulk of that population of viruses, most of them which may not be on a productive pathway. So we wanted to determine when and where HIV encoding occurs. And to do this, Ryan Burdick, a talented scientist in my lab, constructed a vector, and the details aren't important, but he inserted GFP between the matrix and the capsid portion of the GAG protein that Lou talked about, and mutated the cleavage site between them so that it would form a fusion protein, a GFP capsid, and he could co-express it with wild-type unlabeled GAG at a 1-to-10 ratio, so that only about 10% of the capsid molecules would have a GFP tag attached to their N-terminal end. And as a result, these capsids are mostly wild-type, and they can carry out, and they can form infectious proviruses. Their infectivity is similar to that of unlabeled virions. The other modification that we did was we inserted 18 RNA stem loops in the viral genome that specifically bound to a bacterial protein BGL. So when we fuse the BGL to the M-cherry protein, that can specifically bind to RNAs that contain these stem loops. And this was an adaptation of a method that Wei Su's lab had developed with John, anyway, they had developed this in a collaboration with John, actually, at one point in the beginning. But so what we can do is express cells that have this BGLM cherry protein attached to an NLS so that it labels is primarily present in the nuclei of infected cells. And so if there's an integrated provirus that is being transcriptionally active, then that RNA, the nascent RNA that's coming off of that provirus, can become fluorescently labeled with the BGL cherry protein, as shown in this cartoon. And we can then detect that above this background of the BGL signal. So what we did was we infected cells expressing this BGLG protein, LLS protein, infect with the GFP capsid labeled virions. And then 4 hours later started movies and did time-lapse images to capture the infection process. And this shows an infected cell that has a capsid now that's labeled with the GFP that's in the nucleus. So if you can play that movie now. Oh. It just said, yeah. If you can put the cursor on the movie and play it, I could try it here, but yeah, okay. There you go. Okay, so as you can see, a little while later, that signal disappears, and that indicates the capsid has disassembled. And several hours later, we'll see a transcriptionally active provirus appearing very close to the site where that capsid disappeared. And then the provirus expresses a GFP reporter gene that turns the cell bright green. So the disappearance of the capsid signal indicates uncoding, and the fact that it led to this transcriptionally active provirus shows that it's an infectious event, not one of the non-productive events. So that told us that capsids are largely intact when they enter the nucleus. And we measured the amount of GFP signal that's associated with the capsids. And that's shown here in vitro measurement. Compared to the signals in the infected cells, in either HeLa cells or T cells, they have very similar amount of GFP signal, indicating that that capsid must be intact or nearly intact, because we couldn't rule out the possibility that there was some loss of capsid, but we could say that it was largely intact. To determine whether the capsids were intact, Cheng-Li Li, a postdoc in my lab, generated another labeling system where the GFP was fully processed from the GAG. And it also labeled the capsids, but in this case, it was just passively trapped inside the capsid during the virion maturation process. So if the capsid is intact, it's green. But if the capsid is broken, it has holes in it, then the GFP leaks out, what we call content marker GFP. And that tells us about the integrity of the capsid. And what Cheng-Li found was that he could find intact capsids in the infected cell nuclei, indicating that they must be intact. That means we could say they don't have holes large enough to allow GFP to leak out, and that's essentially just one capsid molecule out of 1,500 or so molecules. So fairly small holes would lead to loss of GFP. And that told us that these capsids are indeed intact. So these data tell us that unlike the traditional model, the capsids are intact when they enter the nucleus, and they remain intact until just before integration, near the site of integration. So our next question, just briefly, was what is the molecular trigger for HIV-1 capsid uncoating? And we had found that actually reverse transcription is essential for this. If you block reverse transcription, uncoating is very inefficient. And so Ryan asked, how does this genome size affect uncoating efficiency? And he found that, first of all, the efficiency is correlated with genome size. As the size of the vector increases to close to the full-length genome, uncoating efficiency goes up. But if the genomes are less than 3.5 kb, they really don't uncoat at all. They're not any more efficient than no genome control. So this tells us that you need a certain amount of DNA synthesis for uncoating to be triggered. Now we could take these viral capsids then that have very short pieces of DNA that have reverse transcribed, but they don't create enough pressure to induce uncoating. So we asked, can we disrupt these cores using capsid inhibitors, PF74 and lenacapavir, which then blow up the cores? And if we do that, can that DNA that's inside the capsid go on to integrate and express the reporter gene? And what Ryan found was that if you add lenacapavir, a lot of the capsids, lots of the viral DNA is degraded. So that tells us that the viral capsid, one of its functions is to protect the DNA from degradation by the host nucleases. But what about the few remaining DNAs? Can they integrate and express a reporter gene? So we did a time of addition assay by adding an integrase inhibitor. We found that the remaining DNAs integrate about 77 minutes after uncoating is triggered by capsid inhibitors, and that integration is as efficient as near full-length genomes. So just to then summarize, based on this data, we propose a revised model of HIV-1 replication. We think that after entry into the cytoplasm, the capsids remain intact. They are imported into the nucleus through nuclear pores. Reverse transcription, we think, starts in the cytoplasm, but is completed most likely in the nucleus. And then that triggers uncoating very close to where they integrate, about 1.5 microns or so. And that integration occurs less than an hour and a half after uncoating. So I just told you about these points. And since our publications, there are several labs have confirmed many different aspects of this work, that intact cores enter the nucleus, reverse transcription requires an intact core. And some recent studies have shown that the HIV capsid acts as its own nuclear transport receptor and has actually evolved to get through a nuclear pore, through the diffusion barrier at the nuclear pore. And also conical, apparently intact capsids are seen inside nuclei and docked at the nuclear pore. So based on this, I think I can say that this revised model of the early stage of replication is now broadly accepted by the field. And I wish Howard was here, because I would have loved to know what he thought about it. this work, and I would hope he would have loved it and would have liked it. So just want to thank people in my lab, Ryan Burdick, who did, and Cheng Lee, who did most of these studies, my other lab members, and Wei Su and her lab members, and my collaborators. So thank you so much.
We have time for one or two quick questions.
So if uncoding takes place only in the nucleus, but you can have reverse transcription prior to any of that, are there enough triphosphates in that structure to make the DNA?
Yeah, that's an awesome question, Bill. There are papers that have suggested that the capsid contains pores, like in the hexamer, that form pores, and that DNTPs can get into the capsid through these pores. So I think that solves that problem. Yeah.
Yeah, hi, Vinay. That was great. So if you look at you said 1 out of 50 in the nucleus are infectious, but if you look at the other ones, like, can you tell what happened to them?
We haven't studied that, but I think a lot of them either failed to initiate reverse transcription, or they kind of are misformed and they uncoat or fall apart in the cytoplasm. I think, I mean, this is not our data, but others have reported seeing capsid disassembly soon after entry, and so we assume that maybe a lot of them are defective and they fall apart.
Let's thank the speaker again, and we move on with the. Thank you.